Welcome to our page. I'm Mary Catherine Statham.
And I'm Ariella Tafoni.
And this is our experiment, mutating the 608.1 gene. We began with our parent function, an orange fluorescent gene.
Through this experiment, we wanted to know whether after mutation, the parent function would form a new function, which would result in a new phenotype, or lose its function, which would result in the elimination of the fluorescence.
We predicted that if the 608.1 gene undergoes mutagenesis, then a new phenotype may emerge, resulting in a new function for the gene.
In order to complete our experiment, we began with the random mutagenesis of our parent function, which would create many copies of the 608.1 gene in the hopes that some of the cells have been mutated.
Next, we performed PCR purification in order to remove any impurities that may have contaminated the mutated DNA. Next, we completed a double digestion to insert enzymes into our daughter genes in order to cut them at a certain place.
Then, we cleaned up our digest so that any impurities that may have contaminated the sample during digestion would be removed.
We continued with ligation in order to combine the digested plasmid DNA to the PET15B vector.
Then we performed transformations in order to introduce the BL21 plasmid cells into the ligated bacteria.
We concluded with mini-prep in order to extract the plasmid DNA from the transformed bacterial cells. Once we finished, it was time to analyze our results. In the beginning, we started with our orange fluorescent parent function.
Our results showed that through mutation, the 608.1 parent gene would gain a new function as a duller version of its original self. However, the mutation was not consistent throughout, and some of the mutated DNA lost its function entirely and therefore did not fluoresce.